God's word as true. But if we start with man's ideas as truth, there's a lot of problems and conflicts with what we see today in the field of genetics, and then how it applies to the past. And we'll see that it does not confirm man's ideas as truth. Okay, so I recently gave this a shortened version of this presentation in Oxford, in England. And if I remember, I'll tell you a little story that happened after I presented this, because what I like to do is I'm just going to show you the science. Okay, I'm going to show you the science, and I'm going to show you how it does not support evolutionary ideas. Okay. So let's talk about mutations first. So mutations are really important for evolution because they provide the raw material, basically, the variation in the DNA that natural selection and other mechanisms then act on. So the question is, so we can observe mutations today, all right? So is what we observe with mutations consistent for what is needed for evolution, right? So this is a testable thing that we can look at. So first of all, let's look at some facts about mutations. First of all, mutations only alter existing DNA. Okay? They only alter or change what is already there. They don't originate new things. They just alter current things, alter already existing things. And that typically results in no change or detrimental change. Okay? Like I said before, we think about things like cancer and disease. Now, and sometimes it doesn't make much of a change at all, right? It's just sort of a neutral change. But usually it's detrimental in some way. Now, people ask me, okay, yeah, but what about beneficial mutations? Because that's a favorite of evolutionists. All these beneficial mutations over time have led one kind of organism to become another. But I would argue that there's no such thing as truly beneficial mutations. I would say there are mutations that are beneficial in specific environments that help organisms adapt, but they're not truly beneficial regardless of the context. I would say they're really context dependent. It depends, right? If that mutation is helpful, depending on the environment that the organism is actually in. But nonetheless, even with those beneficial mutations, they don't do what evolution requires. Evolution needs new traits, novel things, right? If you're going to go from some sort of single-celled organism up to mankind today and every other living thing, you have to add stuff, right? You have to add brains and eyeballs and legs and arms, okay? You can't just tweak, right, the DNA that's there, like that say comes from a bacteria, and get a human being. It doesn't work that way. You're going to have to add a significant amount of information. So we're going to look at a couple of examples of mutations that are commonly used in support of evolution. Okay, one is a little more basic, one's a little bit more technical, but to help you sort of understand the problem with mutations for evolution. So the first one we're going to talk about is antibiotic-resistant bacteria, because this is a textbook example of what they call evolution in action. But first of all, let's talk about how antibiotics work. Okay, so when you have an infection, if you are infected with normal bacteria, we'll call them normal, the doctor will give you an antibiotic, and what that antibiotic does is, how it kills the bacteria, is it actually interacts with a protein that the bacteria produces. Now, this is a normal protein, okay, because these are normal bacteria. Now, this protein may be responsible for building a cell wall, breaking down nutrients. It has a function in the bacteria, but it also just happens that it interacts with the antibiotic as well. And when those two things interact, the bacteria die because it causes the protein to not really function anymore and do what it's supposed to normally do. So that's a good thing, right, for us, and that's what we want to happen. But what has happened sometimes, people are infected. I just had a friend actually that this happened to, was infected with an antibiotic-resistant bacteria. These bacteria are mutant. Okay, so when you give someone the antibiotic, it does not interact with the protein because the protein that they produce is mutant. Okay, one of the proteins, whatever it is that happens to interact with the antibiotic, is mutated, it's changed in some way, so the two don't interact, and so the bacteria do not die as a result of that. But why don't they die? Because they've actually become less fit, not because they become more fit. So they are paying a price for this, essentially. So the bacteria, the protein that they normally have is mutated, and so that's why it doesn't interact with the antibiotic, but then it typically doesn't do its normal function very well either, okay? So maybe it doesn't build the cell wall as well, or maybe it can't break down a certain nutrient. Right? Now, sometimes the bacteria can compensate for that, but again, it's still less fit overall, okay? Because it's lost something to gain the antibiotic resistance. So if anything more, it's just a fair trade, shall we say. And we're not But what do you need for evolution? You got to have the gain of traits, right? You got to have new things happening, and this is actually losing something, not gaining something. So I'm going to use the example of Helicobacter pylori. This is the bacteria that typically is responsible for ulcers. Okay, 80% of ulcers are caused by this, and most of the time you have an ulcer, they'll give you antibiotics, it causes the bacteria to die, and you're fine. And that's a good thing. That's what we want to happen. So what happens with H. pylori infection is people will take an antibiotic, the antibiotic gets absorbed through the cell wall of the bacteria, and it interacts with an enzyme, which is another name in this case for a protein, that converts the antibiotic to a poison. Okay, so antibiotics work differently, right? And they work with different proteins. But in this particular case, a lot of times it will interact with a protein that then converts the antibiotic to a poison. Now that's a good thing, because what does that poison do? It causes the bacteria to die, right? And that's what we want, so you can get over your ulcer and get on with life. And so that's a good thing. That's what we want to happen. But because of the overuse and abuse of antibiotics, especially in a lot of third world countries, because they're sold over the counter very easily, there have been mutant strains of H. pylori that are resistant to antibiotics. So when you take the antibiotic, it still gets absorbed through the cell wall. It'll still go into the bacteria, but in this case, the bacteria actually lack the ability to produce that protein that normally the antibiotic interacts with, which then what happens? Well, it can't convert it to a poison because the protein doesn't even exist, the enzyme doesn't even exist, and so they don't die, right? So you still have the infection. So they're an antibiotic-resistant bacteria. So again, just in summary, normal guys produce a bacterial enzyme or a bacterial protein, interacts with the antibiotic, converts it to a poison, they die. Yes, okay? That's a good thing. That's what we want to happen. But with the mutant guys, the antibiotic goes in, they don't produce that protein at all anymore. That normal protein that they normally would produce, they lack the ability to do it because they've had mutation. They don't produce it, it doesn't interact with the antibiotic, it doesn't convert it to a poison, and they don't die, okay? Which is good for them, but bad for us, right? That's not something we want to happen. But again, they've lost something, right? They haven't gained anything. They've lost the ability to produce a certain protein. Now, yes, that might be helpful if these bacteria are living in a hospital or in a nursing home or something like that, where there's a lot of use of antibiotics. Because what that does is it kills off all the other guys, right? So they're the only ones that survive. And they don't have any competition, right? There's nobody around them to eat up all the nutrients in their environment. So they can do it, right? They win because of that. But here's the thing. If you took the mutant guys and you took the normal guys, right? And let's just say they could live in the soil outside your home, and you put them in the soil, guess who wins? The normal guys every time, right? Because they still have that protein. They're still producing it. The protein in this case normally breaks down nutrients. So they have a better ability to do that when they have that particular protein or that enzyme. So the mutant guys are going to die. But in the hospital, the mutant guys win, right? Because all the other bacteria have been killed off, and they're the only ones that remain. So it's only beneficial in a certain environment. It's not beneficial overall, and it's definitely not doing what evolution needs it to do, which is to gain something new, okay? It's acting on an already existing protein. It didn't gain anything new by this to become resistant to the antibiotic. So I always say it's more of a horizontal change, right? It's not a vertical change. It's not evolving into something more complex. It's just a change in the bacteria. So in one research study that was done looking at many different antibiotics and how bacteria become resistant to them, what the scientists noted was that in every case, the bacteria, which you see highlighted in blue here, the bacteria lose something or it's reduced in order to gain resistance to the antibiotic. You see that? What's happening here? So it loses this, but it gains this, right? So there's no net change overall. And again, if you lose something or reduce something, overall it's making them less fit, not more, right? They're getting less fit as a result of that. Again, that's helping them adapt to the environment they live in where there's lots of antibiotics, but overall it's not doing what evolution needs it to do. So it's not a mechanism by which new traits originate, okay? It can only alter traits that already exist. It can't explain the origin of new ones. And that's a problem, and you're going to hear me say this a lot. Okay, because this is important to understand. Because one of the things that evolutionists say, even in the face of this, even in the face of seeing mutations not doing what they need them to do, they'll say, you just have to give it enough time, right? Time is the key. If you just give it enough time, right, you can go from some sort of weird looking, that's supposedly the last, the mammalian ancestor, right? From which all mammals today came, that little purple blob there, okay? So, you can do amazing things, right, if you just give these mutations enough time. And I always say, no, you can't, okay? It doesn't matter how much time you give it, right? You do not have a genetic mechanism to gain the traits needed to go from one kind of organism to another. I am more than happy to give evolutionists trillions of years. They can have all the time they want. But it doesn't matter because they don't have a mechanism. So it's kind of like saying, if somebody said to me, I want you to walk to the end of the stage, but you can't take any steps forward. You can only take backwards, and you cannot go in a circle. I will never get there, right? Doesn't matter how much time you give me. I don't have a way to get there. And that is the same for evolutionists. Mutations do not provide a way. Time is irrelevant. They need a way to do that. All right, now I want to move on to a little more technical example because this is kind of a cool experiment, so to speak, that's been going on for a very long time now. And these are called long-term lines of E. coli. And sometimes you'll see it abbreviated LTTE. And this is in the lab of Dr. Richard Linsky. He's at he's at Michigan State University. And he has been growing these E. coli continuously since 1988. Okay? So that's a long time. It's the longest experiment of its kind that I'm aware of. And he has generated over 64,000 generations of bacteria in that time, more than that. So if you were to, so the equivalent, okay, so these bacteria are being fed nutrients all the time, basically, okay? So in real life, okay, out there in the real world, they're not. They're always having to scrape by, basically, to try to get nutrients. So he's done the equivalent of millions of years in the lab. And guess what? They're still E. coli. They're nothing different, all right? So I think that's kind of interesting from that perspective. Now, he has observed changes in these bacteria over time, okay? That's an observable fact, and we'll talk about some of those changes. I greatly admire his work. I think it's amazing that he's done this experiment this long, and it's pretty cool. So basically what they do is they just keep feeding them, and then once they use up that media, they take it and they put it in another dish with new media, and they just keep doing it. And they freeze the bacteria down at each generation. They'll take some and freeze them down, and then they'll analyze them. So it's really cool. But some evolutionary biologists, like Jerry Coyne, who's at the University of Chicago, think Linsky's work is yet another example, right, textbook example of evolution in action. He said that Linsky's experiment is also yet another poke in the eye for anti-evolutionists. That would be us. The thing I like most is it says you can't get these complex traits evolving by a combination of unlikely events. That's just what creationists say can't happen. Well, that's true. That's what we say can't happen. So, has Linsky's research shown the formation of complex traits via mutation and natural selection? Because this is observable, okay? We can test it, we can look at it and see. We can read his papers and see what he's found. So I will agree that the fitness of the bacteria 64,000 generations later, that they are definitely more fit than the bacteria that he started with, okay? But let me explain. Why are they more fit? They're sort of more fit in a way, okay? Because what we see in the bacteria 64,000 generations out is that they're more fit because certain genes have been inactivated, because there's been a loss or decrease of the function of certain proteins, and there's been the loss of certain regulatory controls. So you know how they're more fit? They're more fit to live in the lab, okay? I always say they've got fat and lazy. So E. coli typically have flagella, which allows them to move, which is good, right? Because they have to be able to find nutrients. They don't just come to them. So they need that. These bacteria lack flagella. They've lost the genes for it. Why? Because they're getting fed every day, right? They've essentially chucked that genetic material, and so they don't need it anymore. So it's disappeared, right? The bacteria that don't have it are fine. They don't need it. And so over time, these things have been lost. So they're more fit. They become adapted to that environment. Now, here's the thing. E. coli do live in the soil. So if you took the E. coli, the parent E. coli, okay, that they started with in 1988, and the E. coli they have now in 2019, 64,000 plus generations later, and you put them in the soil outside your home, The guys 64,000 generations out, they're going to die like really, really quickly, okay? Because they're not adapted to live in that environment. They're adapted to live in the environment of the lab, and that's what they've got, because that's what they've gotten used to. But here's the thing. What do you need for evolution? You need the gain of things. This is not a poke in the eye, okay, as he said. This is not the formation of complex traits. It's every single time. It's the loss of something or the inactivation of something. That is not what you need for evolution. You're just getting alterations in the bacteria. It's not the bacteria on its way to becoming something different than bacteria. Now, one of Linsky's most famous discoveries in his bacteria is his supposedly novel use, and I put that in scare quotes for a reason, of citrate. And citrate is a carbon source that E. coli use sometimes. And one science journalist that was commenting on Linsky's discovery said, "A major innovation has unfurled right in front of researchers' eyes. It's the first time evolution has been caught in the act of making such a rare and complex trait." Oh, that's pretty bold, okay? An innovation, right? A major innovation, and supposedly a rare and complex trait has evolved, right? Has formed. So again, is that really true? Because we can observe it, okay? This isn't something we can't see. We can see it. So normal E. coli use citrate, okay? They already have the ability to use citrate. That's what you need to know, okay? They already had that ability. But they only use citrate when oxygen levels are low, okay, normal E. coli. Now, Linsky's mutant E. coli acquired mutations that allowed them to use the citrate when oxygen is normal, okay? How is this the formation of a complex trait? It's not. It already had the ability to use citrate. It had all the transporters, it had all the enzymes, all of that. The only thing that changed was when it used it, okay? So it's not the evolution of a new trait, but the ability to use an already existing trait under different conditions. That's all that changed, was basically a regulatory control, a regulation change. It just changed when it can use it. Yet, what did the authors of the paper say about their findings? Evolution experiments with microorganisms offer unparalleled opportunities to assess the evolution of novel traits. No, they don't. I mean, you can say that all you want, but when you read the paper and when you look at it, that is not what happened. It already had the ability to use citrate, right? It just now uses it under a different condition. You're not going to get from some kind of single-celled organism to mankind by just changing regulation of things that already exist, okay? You're going to have to do something fundamentally more than that, right? You're going to have to add the ability to have eyeballs and brains and, you know, blood systems and muscle systems and all of that, right? You can't just tweak when it uses citrate and expect to get something else. That's not going to work. So you cannot do that. And so mutation is not, and this is just a small sample, right? But mutation is not a mechanism that allows an organism to gain the novel traits required for movement up that evolutionary tree from simple to complex, right? It only acts on what's already there. It can't originate anything new. And that can be beneficial in certain environments, like we saw with the antibiotic-resistant bacteria, but it's not beneficial overall, right? The overall, the fitness is reduced, so it just doesn't do what evolution needs it to do, okay? And let me tell you, people always think they can get me on this one. They'll send me papers, you know, scientific papers of, oh, look, you know, it's a mutation that added something, and every single time I look at it, this is the result, right? It's a degradation, it's a reduction. It may be nothing more than a trade-off, so to speak. It gained one thing, but it lost something else. That's not helping you get where you need to go. Okay, it's just not doing it. All right, can natural selection, now the next question, which again, these kind of work together, lead to changes at given time produce new kinds of animals? So natural selection is said to select for or against certain organisms or certain changes caused by mutations, leading ultimately to the gain of new things and going up the evolutionary tree from simple to complex. So is that what we observe? Is that what natural selection actually does? Because we can observe this, like I say, in the present. So first what I want to do, though, is define what we mean by natural selection. Because even though it's used synonymously with evolution, they're two very different things. So natural selection, just to kind of break it down, is the process by which individuals possessing a set of traits, we're going to use the example of fur color, that confer a survival advantage in a given environment leave more offspring on average that survive to reproduce in the next generation. So if you want to summarize that really easily, what is that? Survival of the fittest, right? That's essentially what it is in a nutshell. So it has certain traits, it survives better. The idea is it will produce more offspring as a result of that. So let me use the example of bunny rabbits, okay? So let's say we have white, black, and brown bunny rabbits. And let's say they live in an area maybe far north of here, right, where it's very, very cold all the time and there are lots of snow and ice. Who's going to survive? Who's going to make it? The white bunnies, right? Over time, the brown and black bunnies will disappear because they're easier to see, they'll get picked off, and they die, okay? That's just a hypothetical example, but that's how natural selection works. And something, too, I want to set the record straight on is many times people say creationists don't believe in natural selection. Yeah, we do, okay? Because it's observable. I'm going to show you some really great examples of that. It's observable in the present. What we don't believe is that natural selection is the same thing as evolution, okay? Because, like I say, they want to use the terms together, so that's why they'll say we don't believe in natural selection, but I'm going to show you they're two very different things. This is observable, okay? Natural selection is something we can see in the present. Evolution is very different. Because that's the idea that all life on Earth has come into existence by descent with modification from a single-celled common ancestor. This is what we call molecules to man evolution, okay? Goo to you by way of the zoo, you know, that kind of idea. So that's the requirement, but what's required for that? You've got to gain new traits, like I said, as organisms evolve from simple to complex. Now, this is not observable because it's happened over eons of time, right, over millions of years. Here's another argument I see creationists use sometimes. People will say, well, if evolution were true, we'd see it. No, it's a straw man, right? You would not, evolutionists don't expect to see it because it takes too much time, right? They would say it's happening, but it just takes too much time. So we can't observe it. And so natural selection and evolution are two very different things, right? Two very different things. Natural selection is observable. It allows organisms to, you know, adapt and survive within a given environment, but that's very different from evolution, right? Which is one kind of organism evolving into a completely different kind of organism. And yet, like I say, they're often equated, but they're different. So what does natural selection do? Well, natural selection leads, as we're going to see, to the loss or alteration of traits. The name is mutation. What do you need for evolution? You need the gain of new traits, right? Novel things. Natural selection is non-directional, right? It's horizontal change, right? You get variation within or speciation within that particular organism, but you don't get that organism becoming something else, right? It's not directional. It's not going in a certain direction. And natural selection is consistent with creation's orchard, and I'm going to explain that here in a minute. But for evolution, you need a tree. You need to go up, so to speak, the tree of life. So what creationists would say is that God created different kinds of animals, and while we get variation and speciation and change within those kinds, we don't get one kind of animal evolving into a different kind of animal, right? We're not going up an evolutionary tree. We're just got all these different kinds, and we get variation and speciation within those kinds. So the kinds stay kinds. They don't become a different kind, like evolution would require. So let's take a look again at a couple of examples of natural selection used in support of evolution. So one is hypothetical, and the other one is again, we'll see it from an actual scientific journal to help you explain this a little bit more. So we're going to talk about dogs. This is my dog, Harley, okay? He is a Borador, which means he is part Labrador Retriever and he is part Border Collie. So he's like a small lab, basically, which is good, who loves water and loves to herd things, combination of those things. So we know, both in the wild and domestically, that we have many different species and breeds of dogs. And we're going to talk about tonight how we got that. Okay, this is kind of nice. Like one talk builds on the other. I like that. So we'll talk more about that in detail. But how does natural selection accomplish this? Okay, now there's always more to the story. Okay, I'm going to talk about natural selection, but there's also things like genetic drift and migration and founder effect and lots of other things that play a role in this. But we're just going to talk about it with natural selection. So let's say that we have two dogs and they have medium hair. In other words, the length of their hair is in between. So in order to have the in-between hair, you have a gene for long hair and a gene for short hair. Okay, so the combination gives you the in-between. So what are their possible offspring? Well, they could have either short-haired dogs, they could have medium-haired dogs, and they could have long-haired dogs, right? Those are the three possibilities that they could have. Now, let's say these dogs live in a place that's really, really cold, like Siberia, the Arctic Circle, okay, someplace where it's very, very cold. Who is going to survive? Give it enough time, who's going to make it? The long-haired dog, right? The other guys get a bit cold, and eventually they die. Every time I show this, I get that reaction, which is a little scary, but people are like, oh no, the poor dogs. And I'm like, well, I'm sorry, but we live in a fallen world, right? And this is how natural selection works. Sorry, but that's just how it is. It's survival of the fittest, right? So presumably, and this is how populations go extinct. You realize that, right? They get so well specialized and so well adapted for a certain environment, they lose a lot of genetic variety, right? Because what did we lose here? Well, we lost the dogs, but what did we lose? We lost the S gene, didn't we? Right? We lost the ability to produce short hair. We don't have it anymore. And so that's good as long as they stay in that environment, right? To keep producing long hair, because those dogs are better able to survive in that environment. But if all you have is long-haired dogs, then all you can produce is more long-haired dogs. So let's say these dogs, let's say there was a sudden environmental change, or they got transported to Africa where it's very hot. It's going to be a problem, isn't it? Right? The population could completely go extinct at that point because they don't have that S gene. They don't have the ability to produce short hair. Now, some people, I have heard people say, well, what if the L gene mutated and changed to the S gene? All right, that could happen, but they just regained what they had lost before. That's not the gain of anything new, okay, going to some other kind of organism, right? So just gaining what you already lost isn't a change, the type of change needed for evolution. It doesn't help you go from dog to non-dog, okay? It's still a dog. So natural selection, while it can and does lead to speciation in dogs and other living organisms, it doesn't do what evolution needs it to do, right? You're actually losing genetic variety. You're not gaining it. You're not gaining the information you need to go from one kind to another kind. And as I said before, with mutations, time is not the key. And I'm going to say that a lot because it just isn't. People need to understand that. It doesn't matter with natural selection, and it doesn't matter with mutation how much time you give it. You need a mechanism, and you don't have it. People will often ask me, what is the best evidence for creation? Okay, and I'll say, right here. Okay, it's the Bible, right? But if you want to know my favorite scientific evidence, I will say this. You don't have a genetic mechanism. At the base, that's what you need. That's where it changes. That's how you get organisms to be different. And if you don't have it, time is irrelevant, right? You cannot change from one kind into another kind. Now, I want to give you a little more technical sort of real-life example here. And this is taken from an article in the journal Trends in Genetics, which is a scientific peer-reviewed journal. And I really like how it really demonstrated the dilemma between what natural selection does and what evolution requires. And again, I appreciated the honesty of the authors in this article to a degree. So they said this. The title was The Golden Age for Evolutionary Genetics, Genomic Studies of Adaptation in Natural Populations. So the article begins by talking about natural selection and adaptations, how they've been studied in the present. But the very last sentence of the beginning of the article says this: nonetheless, most studies of recent evolution involve the loss of traits, and we still understand little of the genetic changes needed in the origin of novel traits. Yes, right? Because that's what they see. What we're seeing, and it's not really evolution, right? When they say evolution here, they really mean natural selection and adaptation, but they're just, again, they use the term synonymously. They're seeing loss, they're not seeing gain, but that's really what you need for evolution. So they give some examples of natural selection in the paper, and they talk about pigmentation, lightening, and darkening in tetras, a certain type of fish. They talk about wing color pattern changes in butterflies, and they talk about the loss of pelvic spines and structures and armor plating in stickleback fish, okay? And a lot of these are icons of evolution, okay? But again, they're not changing. I always say the butterflies are still butterflies and the fish are still fish, right? They're not changing in anything dramatically new. And these are great examples of natural selection and adaptation of populations to changing environments. I don't know of any evolutionists or creationists that would debate that. Okay, that is a fact, basically. Again, it's observable. It's happened in the present, but there's no gain of new traits or characteristics. There's no direction, right? They're still fish and they're still butterflies. Now, towards the end of the paper, they have a section entitled Origins of Novelty, right? And I want you to listen to what they say. Many of the well-studied examples of adaptive evolution and again, we don't mean molecules to man evolution, they just mean adaptation in the present, have involved trait loss. And they go such as the loss of bony structures and stickleback populations and so on and so forth. However, over the broad sweep of evolutionary time, what we would really like to explain is the gain of complexity and the origins of novel adaptation. Yes, right? That's what you need. You're going to have to do a lot more than just change the pattern on a butterfly if you're going to go from one kind of organism to another. Okay, we got to have some more changes than that. Now, they try to explain it by saying what? There's been a lot of time, right? There's been a lot of deep time here. So it's really hard to study it because it's taken so long. Okay, that's sort of a cop-out answer, but all right. But then they decide, though, they're not going to do that. They're not going to go with the cop-out, so to speak. They're going to try to explain it. And so how do they resolve this problem? They say, well, because natural selection isn't doing what they need it to do. So they say this, and I'm not joking. Of course, to some extent, the difference between loss and gain could be a question of semantics. So, for example, the loss of trichomes, which is a hair-like appendage on flies, could be called the gain of naked cuticle. What? Okay, I'm gonna explain this in easier to understand terms. That's equivalent to a person who is going bald saying, I've not lost hair, I've gained baldness. Does it change? Does it change the amount of hair they have? No, okay? Does it make them sound more positive? Possibly, okay? But it doesn't change ultimately anything. I would say too, it's kind of like a person who's lost all their money saying, I've not lost money, I've gained poverty, okay? True, but it doesn't change your financial situation, right? That's exactly what they're saying here. They're saying we just need to change the semantics, right? We just need to not call it a loss, but call it a gain of loss. That's essentially what they're doing. Now, here's the thing. To an unsuspecting public that doesn't understand this very well, that's enticing, right? And they see the word gain, right? That's what they latch on to. But they're essentially, we need to understand, they're saying the gain of loss, right? You've gained loss is what you've done. But it doesn't ultimately change anything. I don't care how you want to say it. It might make you sound like more positive evolutionist, but it doesn't change the fact that you don't have a mechanism needed to go from molecules to man. So we definitely see the loss or alteration of traits with natural selection, but we don't see the gain of new ones. Just the same, we could say the same thing for mutation. It's non-directional, right? We're staying horizontal here. We're not going from one kind of organism to another. And it is consistent with the orchard idea that God created different kinds, and while we see variation and changes within those kinds, we don't see one kind becoming a completely different kind. So we don't see dinosaurs evolving into birds, so to speak. So natural selection, again, just like mutation, only acts on what already exists, right? And while that may be helpful in certain environments for allowing organisms to adapt, it's not the mechanism by which one kind of animal can change into a completely different kind. So going back to the two original questions that I posed, do mutations add the traits necessary for molecules to man evolution? And does natural selection lead to changes that given time produce new kinds of animals? We can definitely say the answer is no. Right? And again, that's just by looking at the observable science. Right? That's all we did. We just looked at what we can observe today and said, this does not work. You can't take this and extrapolate it to the past and say if you just give it enough time, right, it'll work. It won't, because you don't have a mechanism, right? Time's irrelevant. Time is not the key, and we need to get that. We need to help people understand that. All right, so the third question I want to address is junk DNA really junk? And this has really been a very new and exciting area of research that geneticists have really done over the past 10, 20 years now. So we need to understand, first of all, what junk DNA is. And like I said before, approximately 98% of human DNA does not code for proteins. Okay, only about 2% of our DNA does. And so the other 98% is called junk. That is the scientific term. And so there's many different types. Okay, there's different types of junk DNA, like non-coding RNAs, repetitive sequences, pseudogenes. Okay, that's just some of the categories. So if we were to look at a pie chart of the human genome, only that tiny little thin green wedge there is the protein coding genes. okay, are genes. That's only 2% of our DNA. The rest of it is junk. Now, why is it called junk? Okay. Well, they would say that it's a genetic fossil. Okay, just like we have fossils that we dig up in the ground, right, that are from the past. This is our genetic fossils, or leftovers from our evolutionary path. So Susumu Ono was a geneticist in the 1970s that actually first coined this term, and he said this: "The earth is strewn with fossil remains of extinct species. Is it a wonder, too, that our genome is filled with the remains of extinct genes?" So you can imagine, as you're evolving from some sort of single-celled organism up to mankind, the idea is that over time, just like you've lost certain body parts like fins, okay, tails and things like that, you're sort of mutating and changing the DNA. That's what's led to that. But you haven't chucked the DNA yet. It's still there in your DNA. It's still there in your genome, so to speak, but you just don't use it anymore, okay? So it used to encode information for a tail, or it used to code information for chicken eggs, believe it or not. It used to encode information for all kinds of things that we just don't have or use anymore, and it's just mutated and it's no longer functional. So it's a genetic fossil in that sense. But since the release of the sequence of the human genome in 2003, and discovering that we only had 20,000 genes, there's been a lot of interest in the junk. Okay, so let me just give you a little more background on that. So we produce approximately 100 to 300,000 proteins, okay, as human beings. And so initially, because I graduated the year the human genome was sequenced, okay, with my PhD. So this was a very active area of research when I was in graduate school, and so you would read a lot about it. People thought we're going to have 100,000 genes because they thought one gene equals one protein, right? So they said, okay, we should have 100,000 genes, and that should be the majority of our DNA. Okay? Turns out we only have 20,000 genes, yet somehow we produce 100 to 300,000 proteins. So we're still trying to figure that one out. But, and I've read books on this because I'm a total science nerd, and I like to read books on how they figured out how to sequence the human genome and sort of the technology, how it developed, and the history, and the egos, and all of those things that were involved. And initially, they did not want to sequence the junk. They were not going to do it. They knew approximately where some of the genes were, and so they actually thought to save money and to save time, because all that other stuff's just evolutionary leftovers, we should just sequence the important stuff, right? And fortunately, they did not do that. They decided to sequence the whole thing, but that was the initial thinking. Again, it's because of those evolutionary ideas. So, like I say, though, there has been a lot more research done in this area now, because we realize they're even realizing it can't just be junk. And most of it has been done by ENCODE, which stands for the Encyclopedia of DNA Elements. And it involves many scientists and research facilities around the world, and they have now published dozens of papers in scientific journals. And their aim is to study the junk and to figure out what the junk does. That's their goal. So in 2007, the phase 1 was completed, and they released dozens of papers, and they studied just 1% of the human genome, 1% of the junk, right? So 1% of that 98% basically is what they studied. So it was a pilot study. Well, let's just see, right? Because we do have 3 billion base pairs, so that's a lot of DNA. So they said, let's just study a little bit of it, right? And see if this is worth studying the rest. Well, it was, right? Because what they discovered initially was that nearly all junk DNA gets made into RNA. Okay, so quick biology lesson, or genetics lesson, whatever you want to call it. So DNA, okay, our genes, right, our DNA, and they get transcribed into something called RNA. It's like a copy, okay, of the DNA. And then those instructions get read, and your body figures out how to make proteins from that, okay? So it's like a library book. It's like you're making a copy of a library book in the form of RNA, and then those are the instructions that get read to make the protein. So everybody thought, I was taught this, okay, everybody thought the only thing that goes to RNA is if it's going to be made into a protein, right? Because why bother making it into RNA if it's not going to be made into a protein, right? There's nothing, that's just an intermediate thing, the RNA. That's all it does is make proteins. Okay? We found out we were wrong, very wrong. And it turns out the RNA has a function other than making proteins. A lot of it is involved in regulation. And there's all kinds of RNAs that do this. They have snow RNAs and rRNAs and transfer RNAs and, oh my goodness, so many different types of these things that are doing something just as RNA, even though it doesn't get made into a protein. And so when they figured this out, they're like, oh, well, that's interesting. You know, we didn't expect that. Here were some of the reactions. We are now seeing the majority of the rest of the genome is active to some extent. It's doing something. It's not just sitting there as DNA. We know it's being made into RNA, even though we don't know what that's doing yet at that point. Francis Collins, the head of the NIH, and at that time the head of the NHGRI, he was actively involved in sequencing the human genome. He said, there's a lot more going on than we thought. And my favorite quote, the take-home message is, oh my gosh, this is really complicated. Yeah, I could have told you that, because I could have told you 98% of the DNA was not sitting around doing nothing, right? Because I believe that God designed it. And if it's there, it has a reason and a purpose. Now, I'm not saying 100% of it's doing something. We've had 6,000 years since the fall, right? So there's mutations, there's things that have happened. But I would find it hard to believe that 98% is sitting around doing nothing. And here's the other thing, too. You've got to consider, like I said before, because we're mammals, our genes are very similar to other mammals, right? So what makes the difference? The stuff in between, right? The junk, how it's regulated. We've got to come up with a new term. I'm really tired of calling it junk, but they don't have a new term yet. So phase 2 was completed in 2012. Okay, so 5 years later, they released a bunch more papers, and they studied the remaining 99% of the 98%, okay, that was considered junk. And what did they find? They claimed that 80% is functional because it has specific biochemical activity. That means it binds to something. So now they know not just is it being made into RNA, but where it's binding, okay, where it's doing something, what protein it interacts with. And so they're making these huge databases of this, basically, so that people can study it further. One of the researchers said, almost every nucleotide, every base, every letter of DNA is associated with a function of some sort or another, and we now know where they are, what binds to them, what their associations are, and more, right? And now they have to decode that. And they haven't released a new set of papers yet, and because this is going to take a long time, right? It's going to be, and we may just see individual papers now, but it's going to take a long time to figure out what all those things are. One of the lead analysis coordinators said it's likely that 80% will go to 100%, right? So he's saying the functionality of the genome is at 100%. So like I say, we've got to come up with a new name, because it's not junk, it's not a genetic fossil, it's not an evolutionary leftover, it's doing something. It's clearly designed. This is not the result of random chance evolution, right? Which is what you would expect if evolution were true. You should have a lot of junk, but you don't. Now, one of the things that they concluded, again, they're concluding that the functions are probably regulatory. They're controlling the 2%. Because you got to know when to turn those genes on, how much to produce, under what conditions, you know, the when, where, why, how, all of those things. And so that's what it's involved in. And what's interesting is that we have now found some variations in junk DNA have been linked to disease, okay? Now again, no one ever thought to look there. And one of the claims I hear made against creation science all the time is, well, you guys just say that God did it and that's it. You know, that doesn't open the door to research or more study. And I'm like, are you kidding me? Because God did it and because he's a logical, orderly God, that's why I want to study it, right? And that's why I can study it, and that's why I can understand it. If it's just a result of random chance over eons of time, why study it? There's no logical order to that, right? And that doesn't make any sense. And especially when it comes to junk DNA, because for many years they didn't even want to sequence it, they didn't want to consider it a possibility for disease or the cause of disease, how far behind are we, right? Because of evolutionary ideas in this area. It inhibited science, not creation, not a biblical worldview, an evolutionary worldview did. And so just to name a few things that we know that junk DNA is involved in, there's cancer, there's diabetes, there's schizophrenia, there's celiac disease, there's stroke damage. I have a friend that suffers greatly from Crohn's disease, and they found certain mutations in junk DNA associated with people that have Crohn's, right? So this is exciting, right? This could lead to possible treatments as a result of that. So let me go through a couple different types of junk DNA and then just some other functions that we know of right now. There's non-coding RNAs, which function as RNA. They're not going to become a protein. They're just doing something as RNA. There's repetitive sequences. Now, we talked about that, right, with telomeres and centromeres, so we know that. We'll talk about them more later, but 50% of human DNA basically says the same thing over and over again, okay? But it's not junk, it's functional. And pseudogenes, which are non-functional, okay? They're called non-functional copies of functional genes. They have mutations. They kind of look like another gene we have, but they have mutations in them such that they don't produce a protein, but they're doing something, okay? They are important. So one of the things that this DNA is involved in is gene silencing, right? Because just like you have to be able to turn genes on, you have to be able to turn them off, okay? And so a lot of the controls are believed to be turning genes off. It may fine-tune certain cellular processes, like to go from DNA to RNA to proteins. It's involved in a lot of that. So again, you do it under the right conditions, where, when, why, how, all of that. It's involved in regulating genes during embryo formation. So with some of the DNA, you may only use it while you're growing in the womb. You may not use it when you get older. And I would say as you get older, probably more stuff's being turned off and not working as well as it used to. Yeah, that's part of the degradation, right, of the DNA over time. But they found in certain mouse studies that if they knock out, okay, take out certain parts of the junk DNA, the embryos in the mice do not develop at all. They just stop developing. They cannot develop. Or they're born with severe growth and developmental problems. So tell me that junk DNA is junk again, right? It's not. It's important. DNA formatting. So you have to take 6 feet of DNA and you have to compress it enough that it fits inside a very small part of your cell, okay? And you have trillions of cells. That takes a lot of what we call packaging, okay? So a lot of that repetitive DNA is probably involved in getting it to wind up and twist up so that it can fit inside the nucleus of your cells. It has a function. Even generating genetic diversity in our immune system. I mean, we're exposed to like thousands of foreign agents, okay, like bacteria and viruses and things, and yet we're able to successfully fight most of them all. How is that if we've never seen them before, right? How do we have the ability to do that? It's probably because junk DNA helps to some degree in generating that diversity that we need to do that. So junk DNA is not junk. It really constrains evolution, right? Because what is evolution? Just making, right, random chance mutations in DNA. There's no DNA to mess with because it's all important, it's all functional, it's all doing something. You can't just make changes wherever you want. So in light of this, have evolutionists given up on the idea that junk DNA is not junk? No. Okay, now some have. I think some have, because again, this is observable evidence. And so the ENCODE researchers obviously think that it has function, but some of evolutionists, and usually the vocal ones, are not willing to concede that junk is not junk. So I'm just going to show you 2 quotes from one of them. This is John Timmer. He's a molecular biologist at Cornell and Stony Brook University in New York. And he wrote the following after the first set of ENCODE papers was released in 2007. He said this: "Personally, I fall into the it's all junk end of the spectrum. If almost all of these sequences are not conserved by evolution, and we haven't found a function for any of them yet, it's hard to see how the none of it's junk view can be maintained." All right, so even after they found all this, that it's made in the RNA, he said, I still think it's all junk, because we just haven't found a function for any of them yet. Well, okay, give them time, first of all. And secondly, even at that point, there had been a number of articles produced that showed and published that showed the junk DNA was functional and doing something. So this is a patently false statement, okay? Or he just doesn't read the literature, I don't know. But you can go in literature and find it even before 2007. So that's a problem. So when they released the next set of paper in 2012, claiming 80% had biochemical activity, what did he say? Did he change his mind? And the issue of what constitutes junk DNA is likely to remain controversial. I expect we'll continue to find more individual pieces of it that perform useful functions, but the majority will remain evolutionary baggage. Still not convinced. All right, why? Because it doesn't fit his worldview, right? Again, it's another nail in the coffin, so to speak. It's another piece of evidence that doesn't fit an evolutionary worldview, so he's trying to find a way to explain it away. And I'll be honest with you, to me, and even with all the other things I've explained, when it comes to junk DNA, I'm still a bit gobsmacked when I see these scientists who do very good science every day in the lab. Okay, I'm not judging them as scientists. They do good science. They produce good things most of the time. And yet they will look at this. It's staring them in the face, right? Literally. that this is functional, it's doing something, it's not a genetic fossil, and they say, no, it is, right? And what does that show you? It shows you this is not about the science, right? It's not about the evidence, because that's clear. Romans 1:20 says it will be. It's really a heart issue, right? It really is, because it's a worldview issue. It's a gospel issue. They know that if that's true, then there is a God, there is a creator God, and then what is their relationship to him? And so that becomes an issue for them. So despite what these scientists are saying, there are others that understand this and they get this. And these scientists, who are evolutionists, said, our expectation is that one day we will think of what we used to be called junk DNA as a critical component of truly expert cellular control regimes. Yes, expert, right? Because God is the expert who designed it. So once again, what we observe is consistent with and confirms creation based on God's word and not evolution based on man's ideas of the past. So starting with God's truth makes sense of what we observe in genetics. I would say that's what I love about being a creation scientist, is because I don't have to shoehorn the evidence in, right? It's absolutely consistent with and comes out of a biblical worldview. I say the evolutionists are the ones that are having to make it fit, right? Enforce it or try to explain it away and come up with ideas for it. So if we start with God's word as truth, it makes sense of what we observe in genetics as it applies to the present and to the past, and confirms the truth of God's word, right? Again, because we have a reasoned faith as Christians, we would expect that. But starting with man's ideas as truth, it leads to a lot of problems and conflicts with what we see today and then how it would be extrapolated to the past. So therefore, it does not confirm man's ideas as truth. And again, just by way of reminder, why do we do this? Right? Because we have to be ready to give an answer, and having a good conscience that when they defame you as evildoers, those who revile your good conduct in Christ may be ashamed. Right? We do this because we need to give an answer. We need to tell people of the gospel. We want them to have answers to questions about genetics and geology and astronomy so that they can know God's word is true, right? And it might lead them to a personal relationship with Jesus Christ. And I told you I'd tell you a little story from Oxford, England. So when we were there, we spoke at the Sheldonian Theater. Look it up. It's gorgeous, right? It's a 400-year-old theater. The most beautiful place, no offense, I've ever spoken in. The outside here is very pretty. But, and so it's gorgeous. It's where Oxford University students come when they first come to the university, and it's where they graduate from, okay? So I'm sure Richard Dawkins has spoken there, okay? So we gave a lot of different presentation than Richard Dawkins, obviously. But after the talk, I didn't talk about junk DNA, but I talked about mutations and natural selection to the audience, and Ken Ham also spoke. So he kind of gave the cultural side of things, I gave the scientific side. I had a young man come up to me afterwards, and he said, I'm a student here at the university, and I'm a philosophy student. And I thought, oh, this is going to be interesting. And he said to me, and I could just tell, he's like processing, and he's like, I've never heard this before. And I said, I know. I'm sure you haven't. And I said, so he asked me some questions. He had some questions about like radiometric dating and some things like that. So I kind of walked him through some things and talked to him. And he was very respectful, very nice. And he said, I don't understand how all these scientists can be wrong. And I said, well, you're a philosophy major. I said, it goes back to philosophy, right? It goes back to a person's worldview, right? This really is a heart issue. And I kind of talked to him about some of those things. And he basically said, I'm going to have to go think about this some more. And I was like, yes, okay? And that was great, right? It was one of those times when I was really glad as I stood there in Oxford, right? All these great colleges around and all these PhDs and everything. And there I am in the PhD giving a totally different account of things, right? And it made him think. And that's what I wanted to do, right? I want to make him think about it. And I pray for him. I pray that he finds some good Christians, because I'm sure it's not easy where he's at, that will encourage him. I know that they're there. Will encourage him and help him to know the truth of God's word. So we never know who we're impacting and who we're affecting by being able to give those answers. So I have a couple of just products, genetics, evolution, and creation. I believe that one is back there. Goes through some of these things that I talked about today, as well as some other things dealing with this, because it keeps changing. Obviously, wonder of the cell And the next one I'm going to talk about, Wonder of DNA. They'll be back there later, probably tomorrow. Those will be there. We're bringing those from the States. This is really a walk through the complexity of the cell. This is one of my favorite presentations to give. I have a lot of those. But it talks about the amazing design that we see in the cell, from even looking at proteins that literally walk, okay, inside your cells, which is pretty cool. Wonder of DNA talks about design in DNA. So I do talk about junk DNA a little bit in that one as well. But some other things, hidden codes, and no, not Bible verses, okay, but hidden genetic codes that do things, okay, inside our body that we're finding about. And I talk more about the packaging of DNA and lots of different things. I sort of take a little bit of what I had here and just expand on it a lot. All Creatures, Great and Small, Microbes in Creation, that DVD's back there. This talks about the amazing microbes that we have, okay, in us, on us, everywhere in this room, being inhaled currently. All of those things, okay, those creepy collies, but are really doing very, very good things. And so talk about them in light of creation. And then Replacing Darwin, I talked about that earlier. Great book, again, looking at how mutations and natural selection, from more of a technical standpoint, but how they're problematic for evolution, and Glasshouse as well. And I know a couple of people have said to me, mentioned, hey, I've watched you on YouTube. And we do have a news program that we produce every Monday at 2 p.m. Eastern Time. And it is on Ken Ham's Facebook page, my Facebook page, and the Answers in Genesis Facebook page, as well as the Answers in Genesis YouTube channel. So you can join us every Mondays at 2 o'clock. And what we do, it's usually a panel of 3 of us. And we talk about different cultural and scientific issues that are happening right now, and sort of a biblical worldview interpretation of those things. So feel free to join us. It's a really great time that we have discussing these things. So that's all I have. So I'm going to hand it over to Calvin.